Good. So today we have lecture number 11, which means that we are basically on the home straight. So we managed to get through the 2/3 of the course, and now we are starting the last third of the course, the last 5 lectures, and they will be mostly focused on the level of entire organism or organs, so that we are getting a little bit closer even to the physiology than to the biochemistry. Anyway, we will be considering different chemical reactions, and that's still a biochemistry course, but we are once again changing the level at which we work. So we started with separate molecules, we have considered some organic chemistry, and first 5 lectures, that was basically organic chemistry and what do we do with all that molecules, atoms, and so on. On the next level, so next 5 lectures, we were trying to consider something on the level of cells. So we have considered different I don't know, channels, receptors, some signaling cascades, and so on and so forth. So we were a little bit closer to the cellular level and something nearby molecular biology. Now I'm trying to promote us to even higher level and consider something at the level of the entire organism or organs, or some separate organs. I'm not sure how good have I chosen all that organs, but we should try and look how it goes. So today the lecture on the biochemistry of a muscle. So I will try to consider certain specific reactions to the muscle. Of course, these reactions, some of them will be very specific, so they are contained just within the muscle. Some other reactions may be performed in other parts of the organism, but there should be some logic. Why do we consider them here? Because they have some, I know, some large meaning for this organ and so on. So let's get in. First of all, that's to cheer you up. So 5 lectures left, and we can do it. So let's get to muscle contraction. So here is a pretty simple example. So you all have seen a biceps muscle, and here you see how that looks like from, let's say, a physical point of view. So muscle, they operate by applying tension to their points of insertion into bones, and the bones in turn form various types of lever systems. So if you have studied during some physics class the lever and all that rules, they are absolutely applicable to the joints, to the bones, and so on. Here is example. So when the forearm is bent at right angle, the biceps tendon is attached something around 5 centimeters in front of the elbow. So while the forearm length is something like 35 centimeters. So if you calculate all in all, you should apply a force that is much bigger than the load that you are carrying in the hand, because here the lever works, let's say, in the direction that we probably would not like, but here's how that looks like. You will have a problem for the homework to calculate in more details the force that the muscle should exert on the bone to carry some predefined load, and you will see and you will feel the numbers that we have. Moreover, you will find out which angles are easier to hold a load, at which angles, and at which angles it's more complicated. Here is a few more numbers for you. So the strength of the muscle depends on the cross section, and here you see for the skeletal muscle 0.3, 0.4 megapascal. And here is first tricky question for you. Why do I measure these numbers in megapascals. What do we mean by that? So what's measured in pascals? Pressure. Yes, absolutely correct. In pascals, we measure pressure. And what is pressure? If you think about units of pressure, that's basically, what are they in essence? Well, yes, that's Newton per square meter. So we have force divided by certain area, and that's basically pressure. In this case, we have units of pressure because we are basically talking what? So we are taking some cross-section of the muscle. This cross-section has certain area. If I take the force that the muscle exerts and I divide that force by this area, I will get some number. And the units, the SI units for these numbers, will be basically newtons per meter squared. And these are pascals. So that's basically the reason why do I give you these numbers in pascals. So that's because I have divided the force exerted by the muscle divided by its cross-sectional area. Good. I guess you will use these numbers at a certain point because once again I have there in the homework there is a problem that involves operating with these numbers. So it's good if you understand why do we get these pascals and how are they connected to the cross-section area and to the and to the force. Good. What else? In previous lecture, we have considered the action potential. So the action potential that propagates and so on and so forth. And we were mostly considering how action potentials are propagated to the to some terminal, and then I don't know, we can create action potential in another neuron and so on. But neurons can act on muscles. So each motor neuron that leaves the spinal cord, it innervates multiple muscle fibers. And the number of fibers innervated depends on the type of the muscle. All the muscle fibers are innervated by a single nerve fiber and are called a motor unit. So small muscles that react rapidly and whose control must be exact may have as few as 2-3 muscle fibers per motor unit. While large muscles that do not require some fine control, they may have several hundred muscle fibers in a motor unit. So that's pretty reasonable. So if you have something that you need a precise control, you are trying to control each fiber separately. If you don't need any precise control, you just need some force exerted, and that's all. You probably can collect your muscle fibers together and use one neuron to rule them all. So that's pretty simple. So here you have examples of innervation of muscle with a neuron. Here you see a motor unit, and here another motor unit, and here is a muscle, and that's how it looks. Good. Muscles are connected to bones. Here is just a slide that shows you that sort of connection. So they are interconnecting that bones, and the bones they create the system of levers in our body. So here is example where muscle attaches to the bone. Here you see the tendon. Tendon is basically what connects muscle and a bone. And here you see on the right, you see the structure of the tendon, how that looks like and how it goes. On the left is a little bit more detailed structure of the muscle. We will consider that in much more details in the subsequent slides, but here you see the muscle itself. Here you see the fasciculus and the medium, and here you see the cells of the muscle. They collect together in a single muscle fiber, forming something called syncytium. So they just fuse together into one large giant structure, and this giant structure, of course, since this is a result of fusing few different cells, will contain many nuclei. So here you see, even in this picture, there are 2 nuclei shown, but there may be much more. So they are fused together, and they are performing together all the biochemical reactions. This has to do with skeletal muscles. When we get in the, I guess tomorrow we will get to the heart, and I will tell you that there are something like 2 syncytia, so 2 of them. Regarding other types of muscles that, well, we will get to that. There may be different situation depending on the muscle. Here is the structure of the muscle in a little bit more details. So once again, here you see the bundle of muscle fibers. Here you see these different nuclei in a syncytium, and here you see the sarcoplasmic reticulum. I guess we will get to that pretty soon, because it has a really large role in the muscle contraction. It keeps ions of calcium, and I will show you that ions of calcium, they have a really profound role in the muscle contraction. Here you see myofibrils, and here is one single myofibril. And if you look that myofibril under the microscope, you will see something like here on the left. So you will see different bands, like M band. You will see these sort of Z lines, and all these all these structures. What are they? I guess we will get into biochemical details, but here is an example that shows you what are these structures. So what are they molecularly? So here you have one sarcomere unit. These sarcomere units, they are connected along these the Z lines. So here they are. Here you see the M band at the center, and here you see myosin and actin that slide towards one another. So they are something like this. They are sliding this way, and that's basically the mechanism why the muscle gets shorter or longer. So they can get shorter and exert certain force by sliding one past another. So we will consider today more detailed mechanisms, how it is performed and how are they performing all these sliding, and if we have time, even where do they get energy for this sort of sliding. Good. Muscle fibers can be classified in a certain way, depending on their function, on their biochemistry, and I guess the largest classification gets us to 2 classes. So slow fibers, this is type 1, or red muscle, and the second class will be fast fibers. So easy to guess, we have slow, so we probably should have fast fibers. So slow fibers, they are smaller than fast fibers. They also innervated by smaller nerve fibers. Slow fibers, they have a more extensive blood vessel system and more capillaries to supply extra amounts of oxygen compared with fast fibers. And slow fibers have greatly increased numbers of mitochondria to support high levels of oxidative metabolism, and slow fibers contain large amounts of myoglobin and iron containing and this is an iron containing protein pretty similar to hemoglobin in red blood cells, and it stores some oxygen until the moment that they will need it. So here you see that these muscle fibers, so slow muscle fibers, they rely on oxygenic processes so to get the energy. Here is the myoglobin for you. So here how that looks like. So it's a really small oxygen binding protein and found mainly in muscle tissue, so that's pretty specific to muscles. Of course, you might guess it stores and releases oxygen. It's made of a single polypeptide chain, something around 153 amino acids, of course with the heme containing iron. Its structure is pretty similar to the one subunit of hemoglobin, but it is monomeric. So hemoglobin has something like 4 such subunits, and this one has only one subunit. Pretty tricky question for you. So you remember when we have considered the hemoglobin, there was a pretty interesting effect that we called the cooperativity. So due to this effect, we had that pretty interesting S form of S shape when we have considered the probability of the protein being loaded with oxygen, or some percentage of the sites that can be loaded with oxygen, versus the partial pressure of the oxygen in the surrounding environment. And here's the tricky question for you. Would you expect something similar from the myoglobin? So we had there something like an S shape, if you remember. So there was 100% load, 0% load. Would you expect anything similar from the myoglobin? No idea. In essence, the question is pretty simple. In hemoglobin, we had 4 subunits, and each of the subunits was capable of holding or releasing one oxygen molecule, so O2. When it was bound to one of these subunits, that's promoted or increased the probability that other subunits will bind to the oxygen. Here we have only one subunit. There's no one to promote that probability, so it's not physically attached to any other subunits, so of course we cannot expect this sort of effect. So to have that kind of cooperativity, we need our proteins to be somehow connected, because otherwise how would you promote that? So in case of the hemoglobin, we had different conformations, and so we could that subunits, they interacted, and that's why they were promoting the probability of getting some more oxygen. Here we don't have to interact with, we only have one subunit. Good. And there is some clinical notion that myoglobinuria through presence in urine of the myoglobin indicates muscle damage. For example, rhabdomyolysis. So that's that may be interesting from the medical point of view. Fast fibers, so or type 2 or white muscle. So fast fibers are large for great strength of contraction. Fast fibers, they have an extensive sarcoplasmic reticulum for rapid release of calcium ions to initiate the contraction. So a little bit later I will show you the whole biochemical pathway, how everything is going on, and you will see that things are happening thanks to the calcium ions. Well, not only to them, but they play a crucial role, and without calcium ions you won't get anything. So they are needed. And in skeletal muscles, it's really the main body of these calcium ions are stored in the sarcoplasmic reticulum. So that's why it should be you should really have an extensive sarcoplasmic reticulum, because there is a lot to store in that reticulum, so they really need it. Of course, there are large amount of glycolytic enzymes that are present in fast fibers for rapid release of energy by the glycolytic processes. Please note, the previous type, so the slow fibers, they heavily relied on getting energy from the aerobic processes. So that Krebs cycle we have studied, electron transport chain, all the things they have to do. So they are relying on that processes to get their main share of the energy. That's why they need a very good blood supply, because they need a lot of oxygen. So without oxygen you cannot promote that processes. And of course that's some sort of a bottleneck. So that processes are pretty slow from the biochemical point of view, because you need to carry the oxygen to there, you need to transport all that chemical stuff, so it takes some time, and that's why they were slow. If you want to do things pretty fast, you cannot rely on the oxygen. So fast fibers are trying to rely to some sort of anaerobic processes. So if you need to do something fast, you don't have time to wait for some oxygen to be carried to you, so on and so forth. So you are working with what you have here and now. And here and now you can only do some anaerobic processes, like glycolysis, like making lactate, as we have previously considered, and all that stuff. So all the anaerobic stuff. So that's the deal with the fast fibers. So they will have less extensive blood supply, because they don't rely so much on that aerobic processes. Of course, these processes are you can find them in these fibers as well, but they don't rely on them so much, so they don't expect to have such extensive blood supply as slow fibers. So this sort of oxidative metabolism is of secondary importance. And of course you will expect that they have fewer mitochondria, because mitochondria are you use them to perform all the oxidative phosphorylation and all that stuff, and if you don't rely on that so much, you don't need them so many, so you will have fewer mitochondria. And of course there is a deficit of myoglobin in fast muscle, because once again you don't rely on the oxidation, so you don't need that. Here is the classification of human skeletal muscle fibers, and here you see all that different types. So you see that slow fibers, there's type 1, fibers. So here are certain characteristics of these fibers. While fast fibers, they can be subdivided into types 2A, 2X, and 2B. And here they are. So I'm not getting into the details. You can look after the lecture in the table if you feel interested. What's interesting, depending on how many slow twitch and fast twitch fibers you have, there may be a bit difference which sport suits for you more. Well, of course, if you want to get to some sort of Olympic level, on the on the regular level, it doesn't matter so much. But anyway, here you have a small table. This table shows you the percent of the slow twitch fibers, depending on the kind of sport that you may do. If it's cross-country skiing, you need almost 80%, so you need a lot of slow twitch fibers. While if it's running 100-200 meters, you more rely on the fast twitch fibers, so you don't need so much slow twitch fibers, and here you see that it's less than 50%. And here you see the maximum oxygen consumption that you can correlate with different types of fibers, because you know that slow twitch fibers, they rely on the oxygen, on the oxygenic pathways, on the anaerobic pathways, or on the aerobic pathways, while the fast twitch fibers, they are fast, they have no time to wait for the oxygen, so they rely on the anaerobic pathways, so they do everything. Well, not everything, but they more rely on processes that do not need oxygen. What's interesting, so this sort of relation is genetically determined but varies widely. So elite marathoners like 70-80% slow twitch, sprinters 70-80% fast twitch. It's partially adaptable, so training can shift properties but not fully convert fiber types. So a lot of things, as you see, are genetically predetermined. Endurance training increases the oxidative capacity of fast twitch fibers, so they become more fatigue resistant, something like type 2A hybrid fibers. While strength and sprint training enhances glycolytic enzymes and cross-sectional area of fast twitch fibers. So you see that fast twitch fibers, they can be a little bit modified with training. Complete transformation type 1 to 2 is limited and mostly seen under extreme conditions, so probably you can't get that in the real life. And what can be modified, the mitochondria, capillaries, enzymes, the content of the myoglobin, they are more or less plastic, so even if the fiber type itself is genetically constrained, some biochemical processes may be shifted towards that side that you want. So whether you want to do some sport that needs more of fast twitch fibers or more of slow twitch fibers, so something can be shifted. But you see that genetics strongly influences the sport when you excel, and that's how it looks. Well, of course, if it's going about some sort of Olympic champions, and I guess when you are pushing yourself to the limits of your genetic predetermination. Of course, if you are just doing that not on that high level, probably it doesn't have such a big meaning. Good. Here is one more example. So we have considered all these fibers in the light of sport, but let's take the standpoint of cooking. Why not? Here you see examples that these sort of different fibers, they can be seen if you are cooking some meat. So you probably have heard terms like red meat, that's something like beef, lamb, or duck. It comes from muscles with many red fibers, so high myoglobin content. So fibers with high myoglobin content, you remember that's slow twitch fibers, yeah, because they are slow and they rely on the oxygen. And there is white meat like chicken breast or fish, and they come from fast twitch fibers, which have less myoglobin and more rely on the anaerobic metabolism. And here you see some table with characteristic of meat birds. So here you see the percent of the red fibers, and depending on the situation you may see different colors. So here you see like globin, it's red, and globin protein with the iron oxidated to stage +3, it's brown, and so on. And the thing is, then when cooked, myoglobin changes color. So in raw meat, when where the oxygen carrying myoglobin is red, in cooked meat it's oxidized, and denatured form of myoglobin is brown. So that's why it gets brown. So if meats are cured with nitrite, including corned beef and ham, the myoglobin assumes a stable pink form. So here is example of that pink form. So NO, that's nitric oxide, and a product of nitrite. And in uncured meats cooked in a charcoal grill or gas oven, traces of carbon monoxide accumulate and produce another stable pink form. So you can get pink form either from nitriles or preparing them in a charcoal grill. So that changes the color, so that may be interesting for you. Here is interesting test. I'm not going through all of this. That's a way to calculate some sort of power output of a human by measuring how high can one jump. So I'm not getting into all of that. Here are just some sort of parameters. So the average peak power output measured with this sort of vertical jump protocol averages to these sort of numbers. So here you have this W, that's watts. So these are sort of kilowatts, so 4.6 kilowatts for males and 2.9 kilowatts for females, and here you see the standard deviation. Elite male sprinters can reach more than 20 watt per kilogram peak anaerobic power. That's a lot, and this is anaerobic, so they don't use oxygen for creating that sort of power. While trained endurance athletes rarely exceed 12. So if you are more like doing endurance sport, you will probably not need that sort of high peak power output, because well, they can reach this sort of power, but that just for a few seconds or something like that. And the high power output, this high anaerobic power output, is good for sprinters, jumpers, weightlifters, or hockey players. If moderate and high lactate tolerance, that's good for 400 meter, for example, runners or rowers. While low power output but high aerobic efficiency, that's for some sort of endurance athletes. So if you are doing something that requires endurance. Good. That's some sort of general overview of what we have and what muscles do for us. Let's get a little bit into the biochemistry of what is happening inside the muscle when we are performing contraction. So here you see the sequence of events that should occur for a muscle to contract. So of course everything starts with the discharge of a motor neuron. So that's a command that we need to contract. This discharge releases from the terminal, but releases a transmitter, acetylcholine in this case, that will act on the end plate. This acetylcholine binds to the nicotinic acetylcholine receptors that increases the sodium and potassium conductance in the end plate membrane that generates the end plate action potential. So here you see the generation of that potential. It's a result of increased permeability to sodium and potassium. When this action potential is generated, it spreads, this depolarization spreads along T-tubules. I will show you an image, and I will show you where are these sort of T-tubules, and that causes the release of calcium ions from terminal cisterns of sarcoplasmic reticulum and diffusion to thick and thin filaments. Then it binds to the troponin C, uncovers myosin binding sites on actin. I will show you that as well. And then everything starts working. So the cross linkages between actin and myosin are forming. They start this sort of sliding, and I will show you that in details just in a moment. So here you see the scanning electron micrograph that shows branching of motor axons with terminals embedded in grooves in the muscle fiber. So here they are. On the right you see the structure of a neuromuscular junction. Here you see the neuron is coming, and here is the axon terminal, and when the signal comes, here are synaptic vesicles that contain acetylcholine. This acetylcholine will go into this sort of synaptic cleft, diffuse, and bind to the acetylcholine receptors. So here is sarcolemma, and I guess I have some better view. Here it is. So here you see the action potential came here. For the neuron, as you remember from the previous lecture, we also need some sort of calcium ions. They enter here, they promote that sort of vesicular, they promote the release of the neurotransmitter from this terminal. Acetylcholine is released. Here we have nicotinic receptors that will react to that acetylcholine. So we have sodium entry, so sodium enters. It acts on the voltage, so the potential on the membrane is changed due to the entry of the ions. Of course, this change, this change in the potential, the voltage-gated channels will sense that change in the potential, and it depolarizes. So the adjacent muscle membrane is depolarized to the firing level, and action potentials are generated on the either side of the end plate and conducted away from the end plate in both directions along the muscle fiber. So we are generating action potential. It goes along the muscle fiber, and so the whole muscle fiber will know that we want it to contract, that the neuron sent us its signal, so guys, contract. And then of course we need to clean all this mess up, and the acetylcholine esterase is a really helpful enzyme that will destroy the acetylcholine that we have released into the synaptic cleft, and basically that will be one of the steps of the termination of the signal. So we don't want muscle to be contracted forever, we just need it for performing some work, and that's all. And I guess in the previous lecture I told you that we have certain neuroparalytic gases that basically are targeting the acetylcholine esterase, and one of them was sarin gas used in Tokyo 1995. And you might guess what kind of disaster do we have if this acetylcholine esterase is inhibited and it cannot remove the excess of the acetylcholine, so the muscles, they are constantly getting signal that they should be contracted. So that's no good. So here are some numbers for you that's regarding some release at the neuromuscular junction. So each neural impulse releases something like 60 synaptic vesicles containing around 10,000 acetylcholine receptors. So at rest, small quanta of acetylcholine are released randomly. That's pretty okay. We have already studied that all the processes, all the molecular processes, they are more or less based on some randomness, on diffusion, on some fluctuations, and so on. And I have already told you that if we have any receptor anywhere on the cell, there is always some probability. Let it be very, very small, but there always is some probability that this receptor will be activated on its own, even without any chemicals around, without anything. It will just fire because it's randomness. So it's pretty okay that small quanta are released here and there. The system is built, of course, in a way that it is stable despite this sort of randomness in the processes. If we increase the concentration of the calcium at the end plate, the size of quanta released will be increased. If we increase the concentration of magnesium, we will get some smaller quanta released. A nerve impulse triggers massive vesicle release, and the end plate potential will exceed the threshold and cause the muscle contraction. So that small quanta that are released, they do not cause the action potential on the muscle fibers, so you just don't get any reaction on that sort of randomness. But when it gets to the signal from the neuron, of course, it will fire and we will see something interesting. And of course, if you have some complicated system, you may expect that there will be certain disorders that are affecting this process, and you get some sort of bad disease out of it. And I guess we need to consider, at least very briefly, some of them. So here are two of them, like myasthenia gravis and Lambert-Eaton myasthenic syndrome. There is some shorthand for that, but I don't remember. So here is myasthenia gravis. That's an autoimmune disease causing skeletal muscle weakness and fatigue. Incidence, 125 people per million. And what's interesting, it has some sort of bimodal age distribution. So in 20s, it mostly affects women, while in 60s, mostly affects men. Not sure if anyone knows why. And the major symptom, that's fatigue that worsens with activity. What's the biochemical mechanism? So the antibodies attack nicotinic acetylcholine receptors at the neuromuscular junction. So that's an autoimmune. That's when your own immune system starts recognizing something as a threat. It recognizes something from the organism, and that's really no good. The result, of course, you get receptor destruction and endocytosis of clusters. If receptors are destroyed, of course, the number of acetylcholine receptors will go down, and of course, you will have much lower postsynaptic response to the acetylcholine. So here you see that that's no good, and you just cannot get enough of the signal, so just to tell your muscle fiber to twitch or do something useful. and it is seen even as structural changes, synaptic clefts are getting shallow, widened, or even missing. There are two main forms. One is ocular and the other one is generalized, that influences even limb and respiratory muscles. Fatigue worsens with repeated stimulations and may be associated with different other diseases like arthritis or lupus or something else. And of course, it's often that there is a family history of autoimmune diseases. Well, what's interesting, the treatment here is acetylcholinesterase inhibitors. So I guess pretty interesting turn, I guess unexpected, because what would you expect from the acetylcholinesterase inhibitors? So the first thing that comes to your mind, so probably some sort of paralytic gas. So it's a nasty thing, and we don't need that. But it turns out that in case of certain diseases, these sort of inhibitors can be used at the well, I can say it's a cure, but at least it helps with some of these symptoms. And probably you might get the mechanism. Why does it help? So here's a tricky question for you. Why does this help? So immunosuppressants, that's pretty obvious. We have autoimmune disease, of course we use immunosuppressants to manage that. So it's often associated with some diseases with thymus, so maybe some sort of surgery will help. Okay, why do we need inhibitors of acetylcholinesterase? Nobody knows. Okay, let's get once again. So we have an autoimmune disease that targets receptors and destroys receptors of the acetylcholine. So the number of receptors goes down. Now we have less receptors to fill the acetylcholine. So one of the ideas, so what can we do in this sort of situation? So let's activate these receptors more strongly. So we need a stronger activation for the receptors that are left. So we had 100 of receptors, well, say 20 of them were activated and were performing their job. Now we have only 10 receptors. If proportions are the same, only 2 of them will be activated and we get almost nothing. But if we manage to make a strong signal, we may activate all 10 of them, and maybe that will be not so good as 20, but still may do some job. So these numbers are pretty random, but I hope you get the idea. So if we inhibit acetylcholinesterase, some of the acetylcholine is not destroyed with this sort of enzyme. It still persists in the synaptic cleft, and it helps to activate the receptors because its concentration is higher. So we have higher probability for the receptors to activate, and we activate them more strongly. So that's what we have. So that's the general idea. So we cannot fix the problem itself, but we are trying to patch work, so to find some workaround. So we don't have enough receptors because some of them were destroyed by the autoimmune disease. Okay, let's manage to do something with what is left. Maybe we can activate them. Okay. One more syndrome, LAMS. Uh, Once again, an autoimmune attack, but this time voltage-gated calcium channels at presynaptic nerve ending. So that's decreased calcium influx, and that's decreased acetylcholine release. So that's the problem of the nerve ending at the neuromuscular junction. uh, Targeted approximately 1 per 10,000 people, approximately equally, uh, that's 50-50 men versus women, and clinical features are weakness in proximal lower limb muscles, like waddling gait, difficult lifting arms, and interestingly, it improves with repetitive stimulation. That's because calcium ions are accumulated, and these may distinguish it from the myasthenia gravis. So you see that if you are performing some action repetitively, it may get easier to perform that action. Uh, so it's often associated with some sort of cancer, so the treatment that first of all to manage the underlying cancer, and of course the immunotherapy and aminopyridines that increase the acetylcholine release, blocking some of the potassium channels and activating some calcium channels. While acetylcholine esterase inhibitors have limited effect in this case. Good. Now let's get to our nice pictures, and let's consider in more details what happens in the muscle. So here you have two nice pictures that show the anatomy of a muscle fiber. So how that looks like. Here is the sarcolemma. Here you have sarcoplasmic reticulum. This thing, the thing is called a striate, or here the same striate. That are two terminal cisternae of the sarcoplasmic reticulum. I will show you how they are connected to the T-tubule. So here is the T-tubule. It goes. So here it is. So I promised you to show the T-tubule. So here you see that. And they are connected. There are two types of receptors. I have a nice image in a few slides, so I will show you that. So here is one more slice of all that stuff. So here you once again you see the striate, you see the transverse tubule or T-tubule. So here they are. So here they are. You see the sarcoplasmic reticulum. That's all this blue stuff. Here is the nucleus, one of the nuclei in the syncytium, and here you see all the different myofibrils and bands and Z lines. So we will get to all that molecular details just in a little bit. So just to notice, so during development, myoblasts, so these are muscle precursor cells, they fuse together. This fusion creates one long multinucleated cell, a muscle fiber that can contain hundreds of nuclei. So hundreds of nuclei in one cell. So action potential, that's step 7 on one of the previous slides. So let me so here it is. So here is step 7. So the propagated action potential in the muscle. So here you see the membrane and the action potential that propagates. It travels into the muscle fiber interior through T-tubules. So these T-tubules, they are needed to propagate the action potential to the interior of the muscle fiber. And here are just few notions for you, just to keep record of some terms that we are using. So muscle is an organ, it's made of many muscle fibers. So muscle fiber contains many myofibrils. Myofibrils are made up of repeating sarcomeres, and sarcomere contains actin, that's thin, and myosin, the thick filaments. Okay, we will get to that details just in a moment. And here is the promised T-tubule and receptors that I told you about. So here are two views. This you see the dihydropyridine receptor, and here is the ryanodine receptor. So in skeletal muscle, the voltage-gated dihydropyridine receptors in the T-tubule, they trigger calcium ion release from the sarcoplasmic reticulum via the ryanodine receptors. So they are, in some sense, they are mechanically connected. So upon sensing a voltage change, there is a physical interaction between the sarcolemal bound dihydropyridine receptor and the sarcoplasmic reticulum bound ryanodine receptor. And this interaction gates the ryanodine receptor and allows for the calcium ion release from the sarcoplasmic reticulum. Interesting notice that this connection, this physical connection, is seen in skeletal muscles, but not in the cardiac muscle. I guess tomorrow we will get to cardiac muscle, so maybe we will consider certain properties of that muscle, but just notice for you that this connection is mostly bound to the skeletal muscles. Here's one more picture for you, so that's the repolarization. Here you see that dihydropyridine receptor that is connected to the ryanodine receptor and triggers basically the calcium, that should trigger the calcium release. So here you see how the process goes. So here is the nerve terminal. You get action potential. These receptors, so dihydropyridine, so DHP receptor, DHP receptor acts. It is connected to the ryanodine receptor. The channel is open, and now calcium ions are free to go from the sarcoplasmic reticulum into the cytoplasm. And well, I will show you which processes follow, but this is one of the crucial steps. When the signal is complete, of course, what do we have? So when action potential comes, it's first what? A depolarization. So we are depolarizing, so that's we are going up with the action potential. Then we have a repolarization, so we are going down. Even sometimes we have a hyperpolarization depending on the form of the action potential. So when the repolarization, when it's time for the repolarization, the ryanodine channels are closed, so they just close, and now a special protein works to pump all the calcium ions back into the sarcoplasmic reticulum. Good. Anything else I have? Yes, so SERCA, that's an adenosine triphosphate dependent calcium pump. Good. Here is one more image for you, so that's an action potential propagating along that muscle fiber. Here you see the T-tubules, so here are T-tubules, so action potential is able to propagate inside of that tubules. These thing, that's a triade, so the what we saw on the previous images and what was written down as the triade, so here it is. This is the you might guess that this is the sarcoplasmic reticulum. It has this interesting form. These are the terminal cisternae of the sarcoplasmic reticulum. It contains, as you already know from the previous slide, calcium ions. And these calcium ions will be released when the action potential comes and basically acts through that dihydropyridine receptors and ryanodine receptors. So it acts on the dihydropyridine receptors that are mechanically connected to the ryanodine receptors, which allow calcium to go away from the sarcoplasmic reticulum to the cytoplasm, perform what it performs. I will show you in details just in a moment. And then when the signal is out, it's pumped back with the help of the circa that needs, of course, ATP. So to pump it back, the ATP is required. And here you see the myosin and the actin filaments that will be the target of our consideration through few of the next slides. Here is one more disease I have considered here. That's malignant hyperthermia, MH. That's a hypermetabolic reaction of skeletal muscle triggered by certain drugs in genetically susceptible individuals. So pretty interesting thing, because many people, they don't even know they have this sort of condition, and probably it can be diagnosed only by considering some sort of genetic information or something like that. And that's caused by uncontrolled calcium release from the sarcoplasmic reticulum. The triggers may be some volatile anesthetics like halothane or sevoflurane or something like that. Some depolarizing muscle relaxants. These agents abnormally activate the ryanodine receptor or dihydropyridine receptor and lead to massive calcium release inside muscle cells. The reason is the mutation in the ryanodine receptor type 1, or less commonly in the dihydropyridine receptor. So a small mutation may cause them to react to these sort of anesthetics and massively release calcium that's basically no good. So of course there is some sort of treatment. As a treatment, one uses the ryanodine receptor antagonist. So basically you have that outflow of calcium and you are trying to stop that. How do you stop that? You stop that by blocking these sort of channels that release calcium. And some symptomatic things like cooling the patient is performed. Not so often seen, like one person in 250,000 or something like that, so it's not often seen, and carriers are usually asymptomatic until exposed to triggering agents. So one may not know that it has this sort of mutation, and this mutation may cause such large problems. Of course, you might get why it's called hyperthermia, because if you are releasing a lot of calcium, everything starts like twitching, and of course a lot of energy is lost as a heat, and this may overheat the patient. Good. These are two final slides of the subsection, and here I just wanted to show you some terms. Here is the muscle arranged for recording so-called isotonic contractions. The muscle is fastened to a wiring lever that swings on the pivot. So muscle may change its length, and we are basically recording the length of the muscle under some constant load. While here is the other preparation that's for the isometric contraction, and here you see that we don't let the muscle to change its length, but we are recording the force that the muscle is exerting. That's called the isometric contractions. I guess we will meet these sort of words like isotonic and isometric, so I suggested it's good to write them down explicitly. Here is here are a few more terms for you. So we have considered when just one action potential comes to the muscle, but what basically happens when few action potentials come to the muscle, and what happens. So the term tenderness, so isometric tension of a single muscle fiber during continuously increasing and decreasing stimulation frequency. The dots on the top, so these dots, that's not the image wrongly. That's not the problem with the image, so they should be there, and they signify time intervals, something like 0.2 second. And if we have a single action potential, we get just a single twitch of the muscle fiber. So a single action potential produces one brief contraction and relaxation. But these action potentials, when fired one after another, they may cause so-called the summation of contractions. So stimuli arrive before the muscle fully relaxes, calcium remains elevated, and that produces stronger contractions. So here you see that they are coming more and more frequently, and here you have this sort of summation. There is so-called incomplete tenderness, that's rapid repeated stimuli. They cause contractions to partially fuse with small relaxation intervals. So probably this part is more even close to the incomplete tenderness. And complete tenderness, that's very high stimulation frequency, causes continuous maximal tension with no relaxation phase. So here you see this sort of behavior. So you have some contraction without any relaxation. That's because the signals are coming from the neuron or from whatever stimulatory mechanism you have. So maybe it's preparated somewhere in the lab, that muscle. But if stimuli are coming pretty frequently, you will get a continuous tension with no relaxation phase. The mechanism is basically calcium dependent. So the increased intracellular calcium keeps troponin, tropomyosin complex shifted, allowing continuous cross-bridge cycling. Well, I will show you what that means, what kind of cycling do I mean. And of course, it has a large physiological role, because that allows smooth, sustained muscle contractions for posture and movement. So of course, we pretty desperately need that. Okay, let's get to the contraction on the molecular level. So here is the scheme, once again, of what is happening at the neuromuscular junction, and where are we all going. Here you see the step 1, so the generation of an action potential in the motor neuron causes these small sac-like vesicles within the terminal axon to release acetylcholine that diffuses across synaptic cleft and attaches to specialized receptors in the sarcolemma. So here you see this process of the release. This is acetylcholine. That's all the synaptic cleft, and here you see the acetylcholine receptor. So in this place you see the acetylcholine receptor. And the muscle action potential depolarizes the transverse tubules at the sarcolemma and neuromuscular junction. The depolarization of the T-tubule system causes calcium release from the lateral sacs, so terminal cisternae of the sarcoplasmic reticulum. So here you see action potential traveling along the tube. Here you see the terminal cisterns, and here you see the calcium ions to be released. During the muscle action, actin combines with myosin and ATP, and here you see this sort of mechanism. I guess I have some better pictures a little bit later. So let me move to no, they are a little bit far. So So let's consider first this picture, then we will return to it, reiterate once again and again until we fully understand what happens. Of course, the process of contraction is ATP dependent, and during the muscle action, actin combines with myosin and ATP and also activates the enzyme myosin ATPase, which then splits ATP. And the reaction energy produces myosin cross-bridge movement and creates the tension. So ATP binds to the myosin cross-bridge. This breaks the actin-myosin bond and allows the cross-bridge to dissociate from actin. I will show you a picture of that cycle, so how that dissociates. And the cross-bridge activation continues when calcium concentration remains high enough. So if we have calcium inside the cell, the process will go and go and go. And when the muscle stimulation ceases, the intracellular calcium concentration rapidly decreases as calcium moves back to the sarcoplasmic reticulum through active transport. And the calcium removal restores the inhibitor action of the troponin-tropomyosin. So I guess I will show you these sort of things just in a couple of slides in more details. You will see this troponin complex and different myosin binding sites. So these sites, they are hidden when you have no calcium, but when you have a lot of calcium ions, these binding sites they are released, and you can have binding between myosin filament and the actin filament. Good. But before we get to that, just a small reminder that in eukaryotic cells we have different motor proteins that may act on structural elements such as actin filaments or microtubules and generate movement that allows cells to reorganize their content, change their shape, and even crawl or swim. So if you have ever seen videos with macrophages or something like that, they look pretty cool. And the proteins on which we will focus in this lecture, they move along actin and microtubules, so they can move both, and they cycle between forms having high or low affinity for the filament tracks in the response to the ATP binding and hydrolysis, enabling bind and pull mechanism. So few different families of motor proteins for the eukaryotic cells. So myosins, that's what we will be interested in. First characterized on the basis of its role in muscle, so that's why the name. They move along filaments of the protein actin. Each molecule of muscle myosin consists of two copies, each of a heavy chain with a molecular mass 220 kilodalton, and essential light chain and a regulatory light chain. I will show you that in details on the next slides. And the human genome encodes more than 40 distinct myosins. So some function in muscle contraction while others participate in a variety of other processes. So not only muscles, we have many different myosins. Kinesins have roles in protein, mRNA, and physical transport. Dyneins, they power the motion of cilia and flagella, and a general cytoplasmic dynein contributes to variety of motions in the cells, including physical transport. So here is the motion of a single molecule. So here you see the time and position, how it changes with time. So here you see the myosin V dimer. It's different from what I will show you in the it's a little bit different from what I will show you in the muscle, but anyway gives you some idea of this model. You can see that this dimeric molecule moves in discrete steps along its fiber. Monoproteins have certain structural similarities. So early sequence comparison of myosins, kinesins, and dyneins showed little similarity, but 3D structures later revealed remarkable structural parallels between myosin and kinesin. They both contain P-loop anti-PAse cores, similar to those in G proteins. The dynein belongs to the AA subfamily of P-loop antipases, like in the 19S proteasome, and they have all the heavy chains and so on. Here's more. Here we have in more details what we will be considering. So we are at this stage that we already have our calcium release, and now from all these sarcoplasmic reticulum, calcium was released, so good, and now this system starts working. So here you see some sort of enlarged filaments. So here you see the myosin or thick filament. Here you see thin filaments of the actin. Here is a titin. That's sort of springy protein that, let's say, returns the filaments back to the positions where they were, and keeps them somewhere near the center. All these heads, they are moving, they are cycling between states when they are bound to the thin filaments and when they are released from that thin filaments. So here are a bit more details regarding that sort of mechanism. So here you see the relaxed and and contracted states. So here on the upper part you see the relaxed state, and here you see the contracted state. So that should give you some sort of intuition how that works. So that they are sliding one past another, and that's why you have this sort of contracted state and this sort of relaxed state. So here is the myosin molecule. So here it is. Here is the myosin molecule. Here you see the heavy chains. I told you. Here you see the light chains. You see the head. You see the tail, and so on. I guess we will see some more details in next slides. Here you see the connection between the actin filaments and the myosin filament. So these heads, they are forming sort of cross bridges, and they are connecting to the actin at the active sites. So there are some, I guess, GDP molecules that are bound to these sort of filament, and they are working as active sites for binding these heads. And here you see the troponin complex. So this whole thing is called the tropomyosin, and depending on the concentration of the calcium inside the cell, you have these active sites either hidden or revealed. So depending on whether you need to perform some action or not, you will have these sites hidden or revealed to the heads of the myosin. Here you see how movement is performed. So these are this is the myosin filament, and here is the power stroke performed with these sort of heads. So they attach to the active sites and they change their conformation. So here is some information regarding all that molecules. So I guess I will not waste a lot of time on these slides, so you can look at it a little bit later. Here are a few more images for you to get to the idea of all that myosin thing. So here is the micrograph. So here you see these sort of ATPase domains. So the process uses ATP. I will show you the slide with the process where and how. Here you see the molecule of the myosin. So there is a tail, and there are these sort of necks and two heads. So you see it can be cleaved with certain enzymes like trypsin or papain. So papain may cleave these sort of necks, and trypsin may release part of the tail. treatment of muscle myosin with proteases forms stable fragments, including subfragments S1 and S2, and the light mirror myosin. And each of the S1 fragments includes a head shown in yellow or purple from the heavy chain, and one copy of each light chains shown in blue and orange. And here is the head and the neck, so enlarged view. Here you see the ATP binding pocket, so it will bind to the ATP. Here is the actin binding site, so that's the place where it will attach to the actin. So in this slide we had these active sites, so it will attach to the to that active site with this actin binding site and change its conformation appropriately. So here you see the actin, tropomyosin, and troponin, and how do they all interact. So actin filament, the backbone is a double helix of F-actin, so these are the polymers of G-actin. Each G-actin binds ADP that will become an active site for connecting with the myosin head. The active sites repeat every 2.7 nanometers, and they are anchored in Z disks. Tropomyosin, that's a long protein wound around the F-actin, and in rest it covers active sites, and thus it prevents myosin binding. So myosin in resting cannot seize that active sites, and it cannot bind to this sort of complex. While during contraction it moves, it slides a little bit and reveals basically the active sites. So here you see the F-actin, here you see the tropomyosin, that's a troponin complex. This troponin complex, it moves a little bit. It contains 3 subunits, TNI, TNT, and TNC. TNI binds actin, TNT binds tropomyosin, and TNC binds calcium. And when calcium binds to the TNC, that signalizes that it's time to do something, so its conformation changes and it moves the tropomyosin and reveals all that binding sites. So we can start contraction because now we have where to bind and how to perform something. Here are a few more images regarding sliding filaments mechanism. Guess I'm sorry, I should check how many slides do I have to plan how far can we go. Okay, let's get to this sort of slide. What, that's 49. 49, and we are on 42. Okay, I'm sorry if I take few extra minutes, but that will be a really good place to stop, so let's do it quickly. So here is a little bit regarding that sliding filament mechanism of muscle contraction. I know I still haven't shown you that power stroke with all that heads, but that's ahead of us. I will show you. So in the relaxed state, actin filaments from adjacent V disks, they barely overlap. In contracted state, the actin filaments slide inward between the myosin filaments until they overlap maximally. So the disks are pulled toward the center of the sarcomere. Here you see the plot that shows the length of the sarcomere and the developed tension. So you see there is certain length that gives you the maximal tension that you may have. And here you see how that overlapping looks like. So they are more or less overlapped depending on the on the length. So what drives the sliding? So cross bridges on myosin interact with actin and they generate force. I will show you in a moment how that's performed molecularly. At rest, these forces are inactive, but when action potential comes, calcium is released. All these sort of active sites, where are they? All these active sites are revealed, and everything starts. And here is, at last, here is the slide with the power stroke of myosin in a skeletal muscle. So here you see these sort of tropomyosin complexes, and here is the actin, tropomyosin, and troponin. Here you see the sort of head of the myosin, and at rest, myosin heads are bound to adenosine diphosphate and are said to be in a cocked position in relation to the thin filament, which does not have calcium bound to the troponin tropomyosin complex. When calcium bound to the troponin tropomyosin complex induces the conformational change in the thin filament, and that allows myosin heads to cross bridge with thin filament actin. So here is that sort of calcium release. You see that this calcium release, it exposes the binding sites. So here you see the tropomyosin is hiding the binding site. Now you have calcium interacting with the troponin, conformation is changed, and here you see this active site revealed. So now you can bind your myosin to the active site. So now these heads, they bind to the active site. Since now it's revealed, you see how this sort of conformation changed. So here you see all the binding sites hidden, and here you see conformation changed, and all these binding sites, they are now revealed, and there is a place to bind to. Now the power stroke comes. So myosin heads, they rotate and move the attached actin and shorten the muscle fiber. So that's basically the power stroke. And at the end of the power stroke, ATP binds to a now exposed site and causes a detachment from the actin filament. So here you see the ATP bound in place of ADP. In the image with the with all these molecules, so here you see the ATP binding pocket. Here it is, and that's the place where the ATP will will bind. So here, oops, and ATP is then hydrolyzed into ADP and inorganic phosphate, and this chemical energy is used to recock the myosin head. So here you see the last slide. So once again it's detached and conformation is changed. Here's one more view how that looks like and what are the processes. So you see a scheme for the contraction cycle. So ATP binding to the myosin head dissociates the strongly bound actin myosin rigor complex, and during the recovery step, which occurs with ATP bound, the myosin head can move freely up and down. So it searches for the active site to bind. So here you see the myosin ATP pre-power stroke. So it basically searches for the site to bind. Here ADP and inorganic phosphate are still in that head, but it found the site, connected to the site. Now it's attached and changed its conformation, so we have a power stroke performed, ADP is released, ATP is bound once again, and we are dissociating. And here are the structures as they change during this process. Let me show you. I have found some nice article. So I know it's a little bit counterintuitive what is happening here. So maybe I will show you these images, and maybe they will give you some more intuition on what is happening inside that sort of machinery. So you have like phases. So ATP binding, actin myosin detachment. So tight ATP binding releases myosin from the actin, and then you have a surge phase. Myosin lever arm relaxes from its high energy up position, and the detached head performed a biased Brownian search for a new actin site to bind to that site. ATP hydrolysis occurs during this phase. Then you have rebinding and power stroke. So upon finding a new site, myosin binds weakly, then strongly, and tight binding releases ADP and inorganic phosphate, driving that arm forward. So here is this image, this square shows us all the states that we have, and basically explains us what happens. So here you have the Rieger complex. Upon binding to the ATP, actin is released. So this M, that's myosin, so that's myosin head. This myosin head binds to the ATP, and this drives the release of the actin. Here on the bottom slide, so this big image shows you the energies of all these processes. It's really good because it's a little bit counterintuitive, all these energies and how that looks like. So here you have myosin actin complex. Here you have ATP binding. This ATP binding is really favorable. So you know that all processes go along the way to make the free energy as low as possible. So you want some lower free energy. So now we are lowering free energy by binding the ATP we get here. And you know that in biochemistry, if we have two processes and one needs a lot of energy, we may couple it with another process and spend that energy to drive forward the process that does not want to go forward. And it turns out that releasing the actin is this sort of process. So it really wants to be connected to that binding site. So it doesn't want to leave that binding site. And you need to get energy somewhere to release, to get a release from the actin. And you get that release just by binding ATP. So now we spend some energy. So this is some sort of transition state. So we move through that transition state. It takes something around 1 millisecond. And now we are here. So we have myosin. So you think of that sort of head, and it is bound to the ATP, but actin is now released. So here it is. We are now in this state. Now we transform that ATP into the ADP and inorganic phosphate, but we keep them all together. So this is a pre-stroke phase. You see that that sort of bonds in the ATP, they don't have so much energy. So just this sort of hydrolysis, you see that it barely changes the free energy. Now something interesting happens. We release the inorganic phosphate. So here it is, and bind to the actin. And you remember that releasing that actin, we needed a whole lot of energy to release from the actin. Of course, rebinding back, we get that energy back. So here it is. So here is this part. Now we can do some work. So this energy that we now have can be used to perform a power stroke. So here you see that. So moving from the myosin bound to ADP and to the inorganic phosphate to the myosin bound to the ADP and to the actin, we are performing the power stroke. So here is our work output. So now we have this sort of state. So myosin, actin, and ADP. And now we can release ADP. Here, this is the force transition state. Here it is. We release the ADP and we return to where we started. So we have rigor complex. So that's myosin bound to the actin. So here you see how that works. So we have spent one ATP and we have performed some motion. These processes, they depend a little bit on the load. So if you have a heavy load that slows the ADP release and increases duty ratio, thus slower movement. If you have light load, you have faster ADP release, so longer steps and faster motion. So if there is a heavy load, they all are like waiting for many of that heads to be ready to perform a power stroke, and that's basically performed on the step of of releasing ADP. So whether it spends more time on that in the state with the ADP or it's releasing it faster. And this is the process that we have. Here are a little bit more details. So that's a water molecule that's attacking a G-phosphoryl group of ATP, and you see deprotonated with serine 236. So here it is. So here you see how electrons are moving. So this oxygen is getting this hydrogen while this oxygen is trying to get this one, and here you see what you get at the end. And what's interesting that in effect ATP serves as a base to promote its own hydrolysis. So here you see magnesium, iron, and what's more interesting, these electrons note this arrow. So here you have this bond broken. Know that presented mechanism relies on long-lasting complex of myosin, ADP, and inorganic phosphate. So there should be some evidence of this. Because myosin spends most of its cycle in its primed ADP and PI and inorganic phosphate state. And here is a link for you. That's the article that contains these sort of nice pictures. So you can go there and read even more details if if you need some more explanation. And I will spend just few more minutes. So okay, now we know that the molecule is spending a lot of time in this state where it is bound to the ADP and to the inorganic phosphate. And the interesting thing is that you can prove that. You can prove that. The sort of turnover, like 1 ATP per second, that's unusually slow for the enzyme, and you can prove that something interesting is happening with the experiment with radio labeling. So if you label the oxygen, you can study the processes, and you see that the product phosphate, so ATP hydrolysis, of course, is hydrolysis because water is used, and you use water with heavy oxygen. And the product phosphate contains may contain 2-3 oxygens from water, not just one. So what's interesting that ATP hydrolysis is reversible inside the myosin active site. So it gets back and forth a few times, and during that time it even may reverse some of the phosphates, and that's why few oxygens may end up changed in the products. So that's a pretty interesting part. So you see the enzyme, if you consider the enzyme bound equilibrium for the free ATP hydrolysis, you have a very large K, so the equilibrium constant, but on the enzyme it's only around 10. So you compare 10 to the 5 and just 10. So it's almost equilibrium. And the reason is that enzyme stabilizes the transition state, equalizing reactant and product energies. And hydrolysis itself is not rate limiting, but the product release are rate limiting. So in that in the previous slides, I showed you that releasing some of the products that basically drives the processes forward. Good. And here are some more details. So here is the heavy water, and here you see the ATP, so adenosine triphosphate, and here you see the result in case if you are just performing that sort of hydrolysis, so you will have only one heavy oxygen incorporated. But for myosin, more than one atom of oxygen from water is incorporated in the inorganic phosphate, and the oxygen atoms are incorporated in cycles of hydrolysis of ATP to ADP, and inorganic phosphates and phosphate rotation may be performed. So here you have ATP, here you have water, that magnesium ion. Here hydrolysis is the hydrolysis was performed. In red you see the oxygen incorporated from the water, so if you used heavy water you may distinguish this oxygen from other oxygens. Now the phosphate has rotated, so here you see these. If you compare these two images, you see that red oxygen is in a different place, so it had a little bit rotated, and it may happen that it will reattach once again because you have that K inside of the enzyme close to 10. And once again we can get back to this situation and once again hydrolyze it. So here the water is expelled, but a different molecule of water may come with the heavy oxygen. You perform once again this sort of procedure, and now you will have this oxygen, heavy oxygen, and this oxygen will be heavy oxygen. So you will have two heavy oxygens, and that's basically proves that we have this sort of very long-lasting state when ADP and inorganic phosphate are bound to the protein. And here is just for comparison from the same article. That's a different protein that can move, kinesin. And here you see the two-state and four-step model for the kinesin promoter and the King-Altman diagram. So here it is these four angle. That's the diagram of King-Altman of the kinesin microtubule system. And you see on the bottom the free energy landscape. So you see that here it looks a little bit different. So you have a kinesin, you have ATP bound to the kinesin, and this binding of the ATP is basically used to perform some work. So here you see the power stroke is performed on binding on the ATP. That was pretty different for the myosin, where we had power stroke performed on the binding on actin and releasing the inorganic phosphate. So here you see that on the step 2 you have the hydrolysis, so you get the kinesin with ADP and inorganic phosphate. So here it is, so hydrolysis is performed. Once again we have a really small change in the free energy. Now we release the inorganic phosphate, So that's transition state 3. So here it is, we release the inorganic phosphate. We have kinesin bound to the ADP. And now we need to exchange these, we need to release somehow this ADP. And here you see that it's a little bit complicated to perform, so we spent some energy to do that, but anyway it is transit back to the kinesin without anything bound, and it's ready to rebind to the ATP. That's from the same article, so if you want a little bit more details, so I can advise you to check these articles. So here is the link for you, and you can get some more information. Good, and with that being said, I guess we are done for today. If you have any questions or comments, please. Okay, no comments, no questions. Good. Then I think we are done for today. So have a nice evening and see you tomorrow for the second part of this lecture.